Thank you, Chris, and thank you so much for the invitation to be here. It's such a privilege to be here and see all of the fantastic science that's been happening here for about 250 years, I think it is. So I'm really honored. So today I'm going to talk to you about some of the work that we're doing in my lab, but also speak to you more broadly about the body clock, so the circadian clock, what it is, and why it's important for our health. So to begin with, I'm just going to set the scene by asking you to remember the fact that we live in a highly rhythmic environment. So because the Earth rotates on its axis, the day is a completely different environment from the night. So there's sunshine during the day, it's warm, you can see things, plants photosynthesize during the day, whereas at night it's cold, it's dark, it's a different place in which to be living. And because of the highly rhythmic nature in which the environment keeps changing, all life on Earth evolved with a 24-hour rhythm. So life on Earth really is set to this 24-hour rhythm, and this is because the Earth has a rhythmic environment. So no matter where you look in the tree of life, all life has some form of a clock in it. Whether you're looking at bacteria or insects or mammals or plants, there are rhythmic processes that go on in all of them, such that the day, if you are a plant, is when you gear up to photosynthesize, generate the energy you need, whereas at night you shut down, you respire, and you switch off for the evening. In mammals, in humans, we're active, we're up and about during the day, whereas our rest is for recuperation, the night is for when you reconstitute everything that allows you to then be up and about again the next day. So all organisms possess a circadian clock, and it gives them a major selective advantage in that ability to anticipate those rhythmic changes in the environment, and so to know that in a couple of hours it's going to be daylight, the sun's going to shine, and therefore you should be prepared to do what you need to do during the day, is what provides animals with a huge selective advantage, and that allows them to therefore perform at their best. So circadian clocks have actually been researched for a very long time. So their first description in literature was by this scientist, Jean-Jacques de Marian, and what he described was this plant on the bottom left over here. It's a mimosa plant. It's like a rain tree, if any of you have seen a rain tree, where the leaves open and shut with a 24-hour rhythm. And of course, people have noticed these leaves opening and shutting for centuries, but what he described was that if you isolated this plant and kept it in a cupboard, the leaves would still shut, even if they had no access to understanding what the time was in the outside world. So that was the first description, that there was an innate clock. There was a clock inside the plant that was anticipating these daily changes in the rhythm, and therefore behaving in a way that allowed it to do the correct physiology at the right time of day. And for comparison, what you've got on the top left is a car park, which shows you human rhythmic activity. And on the bottom is a mouse, which is nocturnal. So mice are active at night. And so what you see is that when this car park is active, the mouse is actually sleeping in its nest. But in the evening, it gets on its wheel and is running. So circadian rhythms are really fundamental to our biology. So every aspect of our physiology is really aligned to this outside world. And the job of the circadian clock is to make sure that all of the materials that we need to function are present in the right amount, in the right place, at the right time of day. And so in the human body, the way you see this manifest is that there are rhythms in mood, in growth hormone secretion, our blood pressure. So blood pressure is rhythmic, such that it dips to its lowest during the night and it peaks just as we wake up in the morning. Cortisol, which is the stress hormone, also behaves in the same way, but cortisol is also what is required to get you up and about. And so this is a natural rhythm in cortisol. Of course, the rhythm with which we're all familiar is the sleep-wake cycle, and the sleep-wake cycle is shown over here, where we're asleep at night and awake during the day. But almost every aspect of our physiology has a circadian rhythm. For example, our alertness and mood over here dip naturally to their lowest at night, In fact, the level to which alertness dips at about 2 o'clock, 3 o'clock in the morning, it's to the same extent as if you had had, say, a couple of pints or above the limit to be legally allowed to drive. So the clock has a pretty powerful effect on all of our functioning and all of our physiology. So what is the clock? And what did the science actually tell us in the past about what this clock was and how these rhythmic behaviors were arising? So I'm going to describe some experiments that were done in the 70s, which really sort of kick-started this field. People were looking for a rhythmic basis of physiology and behavior. And these two scientists, Truman and Riddifer, they did some pretty wacky experiments in moths. So they were looking at two species of moths here. On the left is a daytime moth. You can see through its bright colors. On the, which is the Cecropia moth. On the right is the nighttime moth, which is brown, and it's the Pernii moth. And these two moths come out of their cocoon at different times of day. So on the top left, you see the Cecropia, which prefers to come out in the morning, whereas the Pernii comes out at night. So they assumed that the circadian clock had some sort of neurological basis. So they cut off the heads of the caterpillars and then allowed the cocoons to develop. Surprisingly, these still survive. I don't know how. But what you see here is that these moths now lose the rhythm in eclosion. They come out at all hours. Whereas, and you see the same here of the brainless Pernii. And then they wanted to see what sort of signal this was. Was it an actual neuronal connection that you needed, or was there some signal that was just systemic that was coming from this brain? So they took the head of the caterpillar and stitched it onto the abdomen and then let it develop. And again, these things survive. And you can see here, so the loose brain Cecropia now has a rhythm restored, and it starts to come out again during the day, whereas the nighttime one comes out at night. And then they took this one's head and put it in this one's body, and this one's head and this one's body. And you can see over here that now the nighttime moth with the daytime head starts to come out in the morning. So it was really it was a beautiful experiment, and what it showed was that there is a physiological basis to this behavior, and that behavior is encoded within the body in some way that doesn't really rely on environmental signals, right? So yes, I mean, it is still aligned to the light-dark cycle, but whether it's aligned to the day or night is something that's in it, within that moth. So this prompted experiments to then start to look for a genetic basis to the clock. And this field was really pioneered by the scientist shown here, Seymour Benza. So Seymour Benza was based in these labs in California where they were studying fruit flies, and what they were looking for across that space then was genes that specified eye color, wing length, body size, etc. Seymour Benza asked the question whether there were genes that could predict behavior. And behaviors in flies, I mean, fairly simple things. When do they wake up? How much do they fly? How active they are, etc. So he and his graduate student, Ron Konopka, they exposed flies to mutagens and then bred these flies to then see, when you generate a series of clones, whether any of these clones had different circadian rhythms. And then when they isolated these flies with different circadian rhythms, they then looked for mutations that could then be tracked back to certain genes. So this is what they found. And again, this is a rhythm with which the fly now ecloses. So the fly comes out of its cocoon. And what you can see here is that this is a normal fly, which ecloses with about a 24-hour rhythm. These little black bars over here, each line is a day. And you can see these peaks that happen with a fairly regular period, which is about 24 hours. Some of their mutants, though, had completely lost this rhythm, and you can see that over here. Some became really short, so some had now what was a 19-hour clock rather than a 24-hour clock, and some became really long. So this set of flies here had a 28-hour rhythm. What was surprising, though, was that all of these mutations were then localized to the same gene. which he called period. So this was one of the first clock genes discovered and proved for the first time that there is a genetic basis to our behavior. And if there's a rhythm that can be encoded in our genes, there are probably other things that can be encoded as well. So our mood, our memories, all sorts of facets can then also have an explanation in a genetic basis. So this is true of flies. What about mammals? And scientists were asking if the genetic basis for the clock was the same. And then Joe Takahashi was a scientist who then sort of pioneered the same sort of approach in mice, where again he exposed mice to mutagens and then looked at whether he was able to isolate genes. And what he found is that he and a group of other scientists, they found exactly the same gene in mammals as well. So the same period gene or the same PER gene was found both in flies as well as mammals, suggesting this gene has been conserved despite 500 million years. However, when other clock genes were discovered, what they found was that the clock genes in flies and mammals were slightly different. And then if you go further back, what they find is the clock in plants and mammals is almost completely different. Right? Where the fundamental basis is the same. It's a set of genes that allows a loop to occur, but the structure of that loop looks quite different. So what we could make out from that was that the clock was actually something that evolved convergently. And by evolved convergently, we mean, say, for example, eyes over here. The fly eye and the human eye are very, very different structures. They have almost nothing in common, but they serve the same purpose, which is to be able to see the world. So it's thought that the clock evolved in the same way, so convergently, and the only reason for that could have been a sort of arms race, where one species evolved a clock, and in order to keep up, its predator or prey had to evolve a clock so that it could then match its time. So the net result of that is we all have clocks encoded in our genes, and our human clock looks something like this, where you have these two proteins, clock and BMAL, and they're able to bind to DNA. And when they bind to DNA, they can set off an explosion of gene transcription. So they allow these genes to then make products that come out of these genes, and those products can then go off and do functional jobs. So clock and BMAL binding to DNA can then therefore essentially open a recipe book of genes, and then those recipe books are read and produce proteins. And these proteins, in this case we're talking about two of them, called period and cryptochrome. Period or PER, which is the same gene that I showed you before this in flies. Period and cryptochrome can then come back into the nucleus, bind to clock and BMAL, but then when they bind to clock and BMAL, they inhibit them, right? So they stop clock and BMAL from being functional. And so what happens is that clock and BMAL can then no longer produce more period and cryptochrome, which means period and cryptochrome degrade, and then that allows clock and BMAL to bind to the DNA and start that cycle all over again. So that cycle takes 24 hours, and that's how we have a 24-hour clock. And of course, if these four proteins were just cycling between themselves, that would not serve any purpose. But clock and BMAL can, in this way, regulate the expression of thousands of genes. And what you're seeing on the right here is the liver gene expression profile. So each line here is a gene, and its expression is plotted across time. So you're seeing 2 days over here, 2 sets of 24 hours, and red means the gene's going up, green means it's going down. So you can see a whole cassette of genes here cycling, so they go down and then up and then down and then up again. So that is how the clock establishes rhythmic behavior in our bodies. Now this clock is in every cell of the body because it's encoded in our genes, and because it's in every cell of the body, each part of our body is then expressing its profile, essentially reading its recipe book at these times that are established by clock and BMAL, and that's how you get rhythms in our behavior. So in the brain, this results in the sleep-wake cycle, growth hormone secretion becoming rhythmic, blood pressure being rhythmic, etc. So everywhere in the body will follow the same 24-hour rhythm established by clock and BMAL. And this is a really beautiful system which describes for almost the first time how there are genes that can be coordinated to each other and therefore regulate gene expression. But then those genes are also reflecting what's happening in the environment outside because they're behaving with a 24-hour rhythm, which is coordinated with the outside world. So it's really one of the first examples, one of the most beautiful examples of how the environment can regulate gene expression, which then regulates our behavior. And therefore, the Nobel Prize in Physiology was awarded to the scientists who were still alive at the time, who described this process. So Michael Rosbash, Michael Young, and Jeffrey Hall, they were all in some way connected to Ron Konopka and Seymour Benzer, the first scientist I showed you a few slides ago, and they shared the prize. The question my lab asks is, how does this clock know what the time is? There's no point in having a clock unless it's telling you the right time. So how does this clock, which is in our genes, know what the time is in the outside world? And you've all experienced that the clock does rely on these time cues, and sometimes it gets it wrong. So a classic example is jet lag. So when you fly to a different time zone, it always takes you a few days to lock onto that new time zone, and for all your behavior, including your sleep-wake, to then follow that new time zone. And so that also gives you a clue as to what the clock is actually looking for. It's looking for environmental light. So environmental light is really the main time cue that tells your clock what the time of day is. And the way light works is that light during the day, so around midday, has very little effect on the clock. But light at dusk, so the time at which the sun's expected to set, if you see light at dusk, what happens is the clock receives that light signal, and if that dusk signal continues too late, it thinks it's seeing light too late, and therefore it must be ticking too slow, right? So it needs to speed up. It needs to change how it's aligned to the outside world, and therefore it shifts. And therefore light at dusk causes a phase delay, and the next day you wake up slightly later, so then you miss that. On the other hand, if you see light at dawn, early in the morning, what happens is the clock thinks it's seeing light too early, and therefore it needs to compensate, and therefore what happens is the next day you wake up earlier, and therefore you miss that period of light. So the clock is really looking for cues at dusk and dawn to then decide whether it's aligned itself correctly to the outside world. How much light? So this is a sort of schematic of the sort of light we're exposed to in our daily lives. So moonlight measured in lux, and lux is typically a unit that a lot of people, including photographers, would use. So moonlight is typically very dim, 0.1 lux. A candle, which is really the unit by which light is measured, one candle at 1 meter gives you 1 lux. Where we are right now is probably somewhere around 10 lux, 50 lux. Our modern living inside our offices and homes are around this range, about 300 lux. But what we did when we were hunter-gatherers is that we spent a lot of our day at this sort of level, 10,000 lux, 100,000 lux, which is the normal level of light that we're expected to see outdoors. So office and domestic light levels are in this sort of range. But what the circadian clock is really looking for is bright light at this sort of level. And this is because our domestic lights and our office level lighting is designed so that we can see things properly. And seeing requires that our rods and cones in the retina pick up this information and therefore build a visual picture of our world. But the information that's going to the circadian clock is not just from rods and cones, but from another category of cells called retinal ganglion cells, or RGCs. And these RGCs are sensitive to light in themselves, so they're photosensitive retinal ganglion cells. And these photosensitive retinal ganglion cells directly connect the retina to the clock in the brain. And so this sensitivity to light, which comes from this set of cells, only works at higher intensities, which is why we need bright light to synchronize our circadian clocks. So how important is this light, really? And this was studied by a scientist, Ken Wright, about 10 years ago, where what he did is he has a lab at the University of Colorado Boulder, so they're very lucky. They're right by the Rocky Mountains, and he loves camping. So he designed an experiment where he had a bunch of students in their normal office environment, and he put these activity monitors on them, so pretty much like an Apple Watch or Fitbit, recorded their activity and their light exposures for a week in their normal environments, and then he took them for a week camping in the mountains, and still wearing those monitors. And when they were in the mountains, they had no access to devices, no access to artificial light, other than a fire at night. So this is what he saw from their light exposure patterns. So in gray is their exposure during their office sort of environment. So electrical lighting, what you can see here is typical daytime levels reach maybe about 1,000 lux, which is quite lucky. We don't actually get that over here in the UK. But then what happens in the evening is this shoulder. And this shoulder is really interesting because that is something we know exists, but then this was described beautifully in this study, and this is coming really from our behavior in the indoor environment, so evening lights at home, devices, the television, computer screens, etc. But of course, in the natural world, they were exposed to much brighter light during the day, so 10,000 lux, you can see here that this is a log scale, and no light in the evening. So that's their light exposure. What was the actual effect on their behavior? So this is a description of their sleep-wake. So if this black bar over here is their sleep episode. So under electrical lighting, you can see they were all going to bed at about midnight, maybe slightly later, and waking up at about 8 a.m. But you can see the size of these error bars over here, which meant this was quite variable. The time at which they'd go to sleep and wake up every day was slightly different. But after a week of camping, you can see here that their sleep episodes have really consolidated well. There's not much error anymore, and it's advanced. So they go to sleep at about 11 o'clock and wake up at about 7 o'clock. So just a week of bright sunshine was enough to reset the clock and bring it back to where it would have been in its natural state. And this is reflected by measurements of the hormone melatonin. So melatonin is something that is secreted by your pituitary gland to tell you when it's nighttime. It's not a sleep hormone, it's really a marker of the dark. And melatonin, under electrical lighting conditions, you can see is quite variable and late over here, shown in these symbols. Whereas again, after exposure to bright sunshine during the day, you can see how melatonin has advanced. So that's about light itself. And where that light information is coming from into our bodies, it's only from the eyes. People have tried all sorts of things, measured light from behind the knees, from our skin. Nothing else really picks up light information. It's only the eyes. So light goes from the eyes, collected by the retina, and then the nerves from your eyes, the two optic nerves, meet at the bottom of the brain in a region called the optic chiasm. And if you look at the brain just above the optic chiasm, so if you take a slice through the brain like this and look above the optic chiasm, you'll see these paired nuclei on either side called the suprachiasmatic nuclei. Now, each side of these nuclei in mice, there are about 20,000 neurons in these, so they're quite small. But because these neurons receive a direct connection from the retina, from those PRGCs that were telling them how much light they were seeing, each of these neurons can then coordinate a rhythm in that circadian clock, which is then aligned with the outside world. And because this is a neuronal network, it forms a really powerful network of clocks, all of which are wired together. And then the SCN acts as a sort of conductor for an orchestra and conveys this time of day information through the rest of the brain. Which is why the suprachiasmatic nuclei, or the SCN, is called the master body clock. It's the master body clock because its job is to receive light information from the environment and then make sure the rest of the brain, the rest of the body, coordinates to that light environment, keeping time essentially to the same rhythm. This SCN rhythm is located within the brain, but then the outputs of the SCN, which are hormonal, neuronal, behavioral, all sorts of things, that information is then cascaded down into every cell of your body, And when it goes down to every cell of the body, that oscillation in genes that we were looking at before, BMAL and CRY, that oscillation is then timed with the outside world, which is how you get these coordinated rhythms throughout the body of gene expression. And that coordinated rhythm will then give you coordinated rhythms in physiology, which is why we have sleep-wake cycles during the night, growth hormone rhythms, blood pressure rhythms, etc. So our eyes pick up the information, convey that information from the brain to these genes, and these genes are then aligning all of our behavior with the outside world. How we align with the outside world is not the same in all of us. And so this is data from a questionnaire called the Munich Sleep Questionnaire, designed by a scientist, Till Ronenberg, in Munich, where you're asked a series of questions about how, when you like to sleep, what time you wake up, what your preferred windows would be. And what you see here is the data that he collected from thousands of people. And what he found is that there's a normal distribution, right? So we're not all the same. All our clocks are not the same. There are people who are very early, who he called larks. There are people who are very late, he called them owls. And the vast majority of people sit somewhere in the middle over there, and he wanted to call them pigeons, but then he thought doves might be politer. So another thing that had me worried when I first saw this is not the time at which they wake up. This is the midpoint of their sleep, right? This is the time at which they would be in the middle of their sleep episode on a day at which they were free to pick when they went to sleep and when they woke up. So for the vast majority of people, they like to go to sleep around midnight, wake up around 8 a.m., 9 a.m., if they were free to choose their own schedules. So for me, that's already a fairly late type. But what you will see is that normal distribution is a combination of several factors. So it's a combination of your genetics, how old you are, whether you're male or female has a big impact, how much light you're seeing during the day, and when you're seeing that light. So if you see too much light in the evening, that's again going to push you towards this side of the curve. On the other hand, if you see too little light during the day, you're going to wobble all over the place over here. But I'm just going to give you a few examples now for the first three, what genetics, your age, and whether you're male and female are doing. So let's look at age and sex first. So that normal distribution, if we split it by age group, this is what you see. So when you're really young, so young children tend to be early types, right? So again, this is the midpoint of their sleep. So young children tend to have a midpoint of sleep about 3.5 over here. And as they grow older into their 20s, that extends then by quite a bit. But it extends much more in males than it does in females. So males become much later types in their mid-20s than females do. Regardless, both males and females, when they're teenagers and when they are in their mid-20s, are a couple of hours out from much younger children. And then from there onwards, we start becoming earlier and earlier throughout our life course, and through to the point at which when you are in your 60s and 70s, you're back to where you were as a child. So this is a combination of many things, hormonal factors as well. But the take-home message from all of this is there is really a 2-hour difference. So when I am trying to turf my 13-year-old out of bed in the morning, I keep reminding myself there's a solid biological basis to this. They really do need their extra sleep. So let's look at how much of an impact this has, right? This 2-hour difference. So this was a study by Christian Kaioken, a scientist in Zurich, and what he was studying is cognitive performance across the day. So people were asked to do a series of tasks, simple multiplication, code transcription, drawing from a mirror, etc. On the right, you see their circadian rhythm in their body functions, so their physiology, so heart rate, body temperature. You can see these follow a circadian rhythm with the peak sort of midday, early morning. But all of these cognitive processes also follow a circadian rhythm, where again, peak performance is around midday, early morning, right? But now if we put that together with what we just saw, which is where the adult is early, but a teenager is much later, what that means is, again, that should be a 2-hour difference in when teenagers learn best. So some scientists thought about whether or not this means that we're doing ourselves a disservice by sending children to school really early in the morning. So there was a test study done at this school here, Monk's Eaton in Newcastle, where they looked at GCSE attainment. When school started at the normal time, which was 8:50, and when they delayed the school start time to 10 o'clock, such that the students were now learning when their circadian clock was primed to be receiving that information. So the student circadian clock was aligned rather than the teacher's circadian clock. And what they found is that attainment increased, but attainment increased dramatically, particularly amongst the disadvantaged students, when they were allowed those 2 hours extra in bed. Of course, I mean, there are problems with rolling this out. Of course, that means the parents need to go, everything in society needs to change then, right? And that's not a simple issue. So it's still not as widespread as it could be, but there is a thought in there about how we could tailor our own lives and our own behavior to work better with our circadian rhythms. What about genetics? So there are genes, there are variants or mutations that then predispose us to being certain types. So there is a disorder called delayed sleep phase disorder, where, again, these people prefer going to sleep much later and waking up much later. So this is, again, the output from these Fitbit ActiWatch type reporters, where what you're seeing in these black bars is their activity, and when it goes quiet, that's when they're asleep. What you're seeing, this is a control subject over here going to sleep at about 11 o'clock midnight and waking up at about 8. This is somebody with delayed sleep phase disorder, where you can see they don't get sleepy until about 2 o'clock, 3 o'clock in the morning over here. This is actually fairly prevalent. It's there in about 0.2 to 10% of the population, and it turns out to be a mutation in one of those 4 genes that I showed you earlier. So there's a mutation in BMAL and clock sort of drive the expression of many genes, but the brakes, they're repressors, period and cryptochrome. That's where the mutation lies. So if there's a mutation in the cryptochrome gene or Cry1 gene, which then causes it to be more active, more repressive. So when it's more active, what it does is it exerts a stronger repressive effect on clock and BMAL, which makes the clock longer than 24 hours. There's the opposite, which is advanced sleep phase disorder. And again, over here, you can see these people are going to sleep very early. Again, this mark here is midnight. So they go to sleep early and wake up early. And what you can see here is, again, there's a mutation, but this time in the PER gene, so the binding partner of cryptochrome. And the PER gene mutation now makes this PER protein degrade too fast, which means the clock is ticking too quickly, and it finishes that 24-hour cycle much faster than it should, and therefore these people are ready to go to sleep much earlier than they would be otherwise. Here's an example where you see somebody who doesn't follow a 24-hour rhythm, and in this person, it's because they have no eyes. Now, interestingly, the human circadian clock is longer than 24 hours. So all animals that have a clock, very few of them are exactly 24 hours. All of us are slightly longer or slightly shorter. Humans are slightly longer. So if you have no eyes, what happens is the clock exists, but it doesn't get information from the outside world, which means it drifts with time, and you wake up a little later every day. Now, what's happened on this day is maybe somebody in the family or some appointment occurred, which meant that they had to wake up early and tried to reset, but then you can see that drift taking off again after that. So those are specific examples of where you can see disrupted rhythms in society. Here's a more generalized picture of what you would see. So this is advanced sleep phase disorder. This happens in the aged people. In delayed, again, as I said, it happens in teenagers, but it also happens in mental health disorders. It happens in depression, many neurodevelopmental disorders. The free-running condition that I showed you earlier, that's something that you see in the blind, but again, you see it across mental health disorders, neurodegenerative disease. Insomnia, where you have fragmented sleep-wake scattered across the day. Again, this you see amongst the aged, and also disorders that coincide with that group, dementia, neurodegenerative disease. And then, of course, you can have completely short rhythms, which is an extreme case, which you then, again, typically see coinciding with a whole series of mental health disorders. And there's no surprise over here because the clock controls almost every single mental process there is. If you disrupt that rhythm and therefore you're disrupting how it works as a whole, you are going to disrupt all of the processes that go with it, which is why you see this coincidence with mental health conditions. Now, circadian rhythm disruption is not just a mental health condition, it's something that is there in society right now because, again, we evolved tilling the fields. We evolved being in an outdoor environment. But then with the advent of factories, electrical lighting, and now, of course, transatlantic travel, food at any time of the day, devices that we take into bed, really there is no need for us to follow the light-dark cycle anymore. And considering the percentage of the population that is a shift worker or is working late into the night, the estimate now is that 30% of the population has circadian disruption of some sort. And what are the consequences? Because the clock, of course, controls all of our physiology. There are many consequences: a suppression of the immune system, an increased risk of metabolic disorders, and of course a whole raft of psychiatric disorders. So I'm not going to talk about the psychiatric side today, and I'm happy to chat about this later. I'm just going to give you an example of why you would see an increased risk of infection and an increased risk of metabolic disease. So this study, which is a really nice one, and this was their sort of cover, was you can use the clock to harness essentially those processes that are rhythmic to make sure you are eating and therefore metabolizing your food in a way that works with the circadian rhythm and prevent weight gain. So these two groups of mice were both fed a high-fat diet, so about quite a lot of fat, like a burger and chips meal. And what happened was that both groups could eat that high-fat diet meal, but as long as you restricted that meal to when the clock was prepared to receive it, you could prevent weight gain. So as long as you're working in synchrony with the clock, you can actually make things much better than they would be. So to restrict those high-fat, high-sugar meals to the day for us, preferably breakfast for us, would be the best thing that you could do. So for example, in the liver, here is, I showed you all of those rhythmic genes earlier in the liver. What are those rhythmic genes? Those are genes that are involved in glycogenesis, in gluconeogenesis, in lipid metabolism, every process. You name the metabolic process in the liver that is going to be regulated by the circadian clock. And this study showed this really nicely, where on the left over here, what you're seeing is where people in this study group were fed identical meals, right? So they were fed identical meals for breakfast, lunch, and dinner, and how much their blood glucose shot up after every meal was then measured. What you can see here very clearly is that although they ate the same thing, the level by which the blood glucose shot up was much higher for the evening meal than it was for the morning. So again, eating that high-fat, high-sugar meal should be restricted to the morning. And then what they did is to provide a constant infusion of glucose, just to see, well, is this really to do with the meal or is this to do with how your body is processing it? And so even with a constant infusion of glucose, what you can see is the glucose infusion at night results in much higher levels of blood sugar during the night. Similarly, over here, if you compare shift workers with normally working people, what you see is that the level to which the hormones that regulate hunger and satiety, they're completely deregulated. So they follow a completely different pattern in these people who are misaligned and shift work type people compared to those who are maintaining a normal rhythm. I don't have time to go into this, but again, I'm happy to chat later. Another one is where the biobank, which is this huge repository of human genome data from the UK, but also alongside a huge amount of phenotypic data, life history, medication, brain scans, all sorts of things in the biobank. What they found from analyzing these data is that shift workers were more likely to be hospitalized with COVID than other groups. So if you were unlucky to contract COVID, if you were a shift worker, the chances are that you would have had a more severe outcome. So we asked, why is that? Why would that be the case? And so what we did is to collect lung tissue after giving them acute sleep, so taking away their sleep, disrupting their clock, for mice, not humans, and then saying, what changes in the lung if you disrupt the clock? What we found is that the genes that changed in the clock, surprisingly, again, these was healthy mice, right? There was nothing wrong with them, but they were genes that were already implicated in viral infection, so HSV infection, HIV infection, because those were the things that were well described at the time, not COVID, which told us that if you were not sleeping well, the pathways that were being affected in your lung were to do with infection and immunity. And then we said, well, how much of this actually impacts on how COVID, how the SARS-CoV-2 virus would therefore be using that landscape, as it were. And what we found is that about a third of the genes that CoV-2 relied on for its survival were also regulated by your sleep history. So whether or not you slept well was impacting a huge portion of what SARS-CoV-2 needed in order to replicate. In a bad way, right? So for example, over here, you would have all heard of the spike protein. That was what was targeted for the vaccine. So the enzymes that are responsible for spike protein cleavage is one that's regulated by the clock. And every stage of the life cycle over there, from what happens to the virus when it goes in, how it replicates, how it's released, all of these are regulated by the circadian rhythm and how well you've slept, which is why sleep and circadian rhythm can change the landscape in your lung in a way that predisposes it to be more receptive to being infected. So another angle of what my lab is doing is to say, well, what do we do about this, right? So we know that the clock is regulating a lot. We know that circadian disruption is a fact of life. How do we fix a clock if it's broken? What we do at the moment, including me, is you set an alarm clock to wake up. First thing we do is look for tea or coffee or a cigarette, and that wakes us up rather than the circadian clock. And then at night, we all rely on a glass of wine or sedative hypnotics to put us to sleep. That's not the way we should be doing it, because none of this is actually fixing the clock. None of this is actually working with the physiology that we need to be working with. So in my lab, we've been thinking of ways in which we can then go in, understand these pathways, and therefore fix the clock. For this, the main system that we've been thinking of is to try and copy what light does, right? Because light is the most important signal to the clock. How does light speak to the clock? And can we copy those pathways to then make new drugs, for example? So by understanding those pathways, we then get a handle on how nature shifts the hands of the clock, and therefore can we copy that and make ways in which we can shift the hands of the clock. So what does light do? So what we're studying in my lab is when that light information is picked up by the eyes and sent to that SCN, what is the SCN doing with that information? So what are the molecular signals like at that stage, which neurotransmitters are involved, and how does that molecular information eventually converge on clock genes and change their expression? For example, one study that we did in the past is to say there must be some reason why the effect of light on the clock doesn't go on forever. And that answer is probably there if we try and understand what's happening in jet lag, because we've all experienced this, where if you fly to a new time zone, if the effects of light on the clock were not limited, we would all be there in the new time zone the next day as soon as we saw the sunrise. But that doesn't happen. We take several days to adjust to a new time zone. So what we did is to expose mice to light at a time at which they are not expecting it. So light at night, so this is, for example, if they had taken a flight west, they were seeing light at night. And then we took the SCN and looked at the gene expression landscape of what was changing. We found lots of genes, but I'm just going to condense what we found down to the fact that light information goes in through neurotransmitters and switches on the expression of this clock gene, PER1, which is the same one that we met at the very start of the lecture. And PER1 is what goes off and shifts the hands of the clock. But alongside PER1, what we found is that there was also another gene switched on called SIK1, or salt-inducible kinase 1. And the job of SIK1 is to add a stamp onto this factor that is responsible for the production of PER and switch it off. And therefore, SIK1 can act as a brake on how much the clock can shift. And so this is the experiment that we did. So you take we took mice, and we flew them east. So we shifted their time zone by 6 hours. And what a mouse would do is normally it would be on this activity bar. So mice are nocturnal, so the gray bar is its activity. They're active at night. And then if you shift the light-dark cycle 6 hours, what happens is the mouse will move its activity by an hour each day until it locks onto the new light-dark cycle. And that is what we would do as well. But if you remove SIK1, which was the brake on this, what happens is that that same light information is now no longer limited, and the clock can move much further, much faster. And what you can see is that the mouse now can re-entrain in 1 day. So this is a schematic, and these are the data. So these are some of the first data that I generated when I was first doing research in this field. So I still show this every time, although this was published in 2013. You can see here, this is our control mouse where you can see its activity. Again, each line is 1 day, and the gray is where the dark is setting off. So that is when the mice are active. You can see here we've shifted the light-dark cycle, and the mouse moves its activity a little bit each day following this red line. But if you remove SIK1, what you see then is that the activity jumps. And by the next day, this mouse is now adjusted to the new light-dark cycle. So, and we're taking this further now in my lab and trying and understanding what SIK1 does. But what we're also doing is to ask, with an understanding of these pathways, is it possible to drug them in a way to fool the clock into thinking it has seen light? So for this, what you can do is express the clock in, well, the clock does express in every cell, but you can track it. You can track it using luciferase, which is the enzyme that fireflies make to glow at night. And so by tagging the clock genes with luciferase, what you see are these rhythms and glow in the cells, and then you can image these cells in anything, a microscope, a reader for light levels, and then what you can see from there is how the clock is oscillating. Now that same rhythm and glow you can recapitulate within the SCN, so within the master clock, and then you can also look at what happens to the behavior of the mouse by putting the mouse in a wheel. So we get the same readout from cells all the way through to behavior of understanding what the clock is doing. So this is sort of what the data look like, where you see these cells have been recorded for 5 days, and you see 5 days of genes going up and down and up and down and up and down. And you can look for any treatments that might make this rhythm stronger or weaker, longer or shorter, or shift it such that its alignment is now moved with the outside world. What we saw is that adenosine receptor antagonists were very strong resetters of the circadian clock, or they were able to mimic that effect that light was having really well. Now, you're all actually familiar with adenosine receptor antagonists because probably the drug that is most widely used in the world is caffeine. And we use caffeine to stay awake. And how does it work? So what happens in our bodies naturally is that we build up a metabolite of our energy molecules called adenosine in the brain. And adenosine, when it binds to its receptors, it signals sleepiness. So caffeine is an adenosine receptor antagonist, which means that it can block those receptors from sensing that sleepiness drive that was coming from adenosine. So when you take caffeine, you feel more awake because you're not allowing that sleepiness drive or adenosine to work. So which is why when you take caffeine, you feel wide awake, but at night you avoid caffeine and therefore allow adenosine to bind to these receptors and kickstart sleepiness again. Now, if it's true that adenosine receptors were resetting the clock, caffeine should be doing it. And there was evidence in the literature that caffeine was able to do this. So this study showed that when subjects were fed a double espresso at bedtime, what happened is that the clock shifted, right? So the clock delayed by an hour in response to caffeine. So not just their sleep, but actual measurements of their circadian rhythm were shifted out by an hour. If you combined that caffeine with light, it had a bigger effect. So if you were seeing bright light in the evening, so if you were watching a bright screen and you had a double espresso at night, the effect on your clock would be shifted by about 2 hours. And so caffeine does have an effect on the clock. So we tried to see whether this was true of specific adenosine antagonists as well. So we were studying a few in the lab. And what you can see here is we saw the same thing, which is that this drug, an adenosine antagonist, administered to mice at dusk. So dusk is the time at which the clock will delay in response to a signal. You see the clock in this case has jumped. The two red lines don't meet after the red triangle, which is where we gave it the drug. So the clock has moved in time because it thinks it's seen a signal like light, which has told it to move, except that in this case it wasn't light, it was the adenosine receptor antagonist that we gave it. Similarly here, we did the jet lag experiment, which is where we asked the mice to wake up 6 hours earlier. And you can see here in the beginning they would struggle, but with an adenosine antagonist or with a coffee, they would be able to entrain much faster to this new light-dark cycle. So the question that we asked as scientists was why? Why does adenosine regulate the circadian clock? Yes, adenosine regulates sleep, but shouldn't that be kept separate from the clock? Well, the answer turns out that what adenosine is doing in this situation is switching off clock genes in a way that tracks sleep state. So in this situation, what this means is that how well you've slept can regulate how much of an effect light has on the clock. So if you remember this graph that I showed you earlier, light at dusk will delay your clock. It causes your clock to move later in time. And this is what you see here. So mice, when you expose them to light at dusk, they typically respond by shifting their clocks by about an hour and a half. But if you do the same experiment after sleep depriving the mouse, so not allowing it to sleep, and then you shine the same amount of light at it, the clock can't shift to the same extent. The clock can only shift by an hour. Now, if that had anything to do with adenosine, if we blocked the effect of adenosine with something like caffeine, in this case, an adenosine receptor called J&J, we were able to restore that effect of light, showing that it was adenosine that was mediating the effect of sleep on the circadian clock. So we thought that adenosine is providing information on your sleep history to change how your circadian clock can perceive light. Now, why would this be useful? Where do we see this in nature? So typically where you see something like this is where you compare how nocturnal and diurnal animals use light information. So in mice as well as humans, mice being nocturnal, we have the same circadian clock. It ticks in the same way. The only difference is that that information from the clock in mice is telling them to go to sleep. In us, it's telling us to wake up, right? How the clock receives light is again same in both nocturnal and diurnal creatures. The only difference here is that for nocturnal animals, it's evening light that has a big effect on their clock, whereas for us as humans, it's morning light that has a big effect on our clock. So why is that? It could be because while mice are awake throughout the night, when they wake up, it should be the other way around, sorry. So they're asleep throughout the day. So when they wake up at night, the levels of adenosine are low, which means the level of inhibition on the clock is low, which is why light can have a big effect at dusk. Whereas for us as humans, we've been awake throughout the day, which means the levels of adenosine at dusk are high, and so the level of inhibition on the clock is also high, which is why it's light in the morning that has the bigger effect, not light in the evening. So we decided to test this. So over here, we used adenosine drugs that act on the excitatory arm in orange, and we hypothesized that they should make the effects of light on the clock bigger, whereas in pink over here, we used the inhibitory arm, which should make the effects of light on the clock smaller. So again, it's the same amount of light, but with the drug, we should see a bigger shift at dusk and a bigger shift at dawn if the drug is orange, or a smaller shift at dusk and a smaller shift at dawn if the drug is pink. So these are the results from the experiment, and that's what you see. Orange drugs make the shifts bigger, whereas pink drugs make them smaller, which was evidence that adenosine acts directly on the light input pathway to the clock. And what this meant was that adenosine, acting on its antagonists, can piggyback on the same sort of mechanisms that light uses to communicate to the clock. And therefore, what the animal does by summing up the information from your sleep history coming from adenosine and light coming from your eyes is that it can decide based on how much it has slept and how much light it's seeing, and when it's seeing that light, what the right time window is for it to be asleep. So the clock sort of combines information from different parts of our experiences and decides what to do with it. So sleep history can modify the response to light, but the flip side of this is, of course, that you can drug adenosine antagonists, right? If we have caffeine, there are multiple other things that are specific and probably much safer to use that can target the adenosine receptors, and therefore we can use them as drugs to target the clock. So where we're hoping to use these are in the blind. Again, these people have no light, and they have no way of perceiving light, rather. And therefore, in this population, if you're able to give them something that's able to fool the clock into thinking it's seeing light, you can restore a sense of time. Again, where you see this is in eye loss, but then it is applicable in shift work, mental illness, a whole range of conditions. Again, we're hoping to work with the blind veterans, or a population that has lost their eyes due to trauma in battle. And this is our pilot experiment in mice. Mice, you don't need to take their eyes out. You can just drop them in darkness. They can't see then. And so you can see here a mouse's clock is shorter than 24 hours. And so it wakes up a little earlier every day. So that's the black bar there. Its activity is a little earlier every day. But it still runs with a circadian rhythm, but that rhythm is not 24 hours. It's about 23.5. But if you do the same experiment by giving it a dose of that adenosine antagonist every day over here, you can see that it's now locked onto a 24-hour rhythm instead. And we showed then that this is dose responsive and therefore something that could, in theory, be used in the clinic. So I'll end on that and thank everyone in my lab involved. So the data that I showed you today were generated by Zainab, Simona, Kwang, and Lewis. And all of the drug work was done in collaboration with Sri Vasudevan, who's also at Oxford, and Russell Foster, who's been a long-term mentor of mine. So I'll stop there and thank you, and thank my funders and the RI as well.